Hello dear students, welcome to GDC. In this video, we are going to talking about model paper 3. This model paper is specialized designed for Telangana Pharmacist Exam based upon the latest syllabus. So in this video, we will talk some 20 more MCQs with the detailed explanation. Let's move to the first question. Yeah, which of the following is an example of nucleosides? Okay, which of the following is an example of nucleoside? Your options are FAD, NADP, CAN, yeah, adenosine. Yes, the right answer is your adenosine. Adenosine is an example of nucleoside. So what is nucleoside? So nucleosides are, they are basically, nucleosides are contains nitrogenous bases. Okay, nitrogenous bases such as your purine and pyrimidine and a pentose sugar. Okay, however, if we'll talk about nucleotide, then it contains your nitrogenous bases plus pentose sugar and your phosphates. Okay, so if we'll see the nucleoside is a monomer of nucleic acid like DNA and RNA, whereas nucleotide is a monomer, whereas nucleosides are nucleoside bases linked with a pentose sugar molecule. Nucleotide, pentose sugar, nitrogenous base and phosphate group. This is only nitrogenous bases and pentose sugar. They polymerize and form a nucleic acid, as well as they act as a energy providing sources. Nucleosides are good anti-cancer compound, as well as they have a antiviral property. Example of nucleosides are your cytidine, uridine, adenosine, guanosine, thymidine and inosine, whereas nucleotide example is your deoxyribonucleotide, ribonucleotide, ATP, NADP, FAD and CAM. Okay, so moving to the next question. A uncompetitive inhibitor binds to, there are two type of or basically we can say there are three different types of inhibitions. Okay, one is a reversible inhibition, second one is irreversible inhibition, third one is your allosteric inhibition. So reversible again divided into three subclasses. Okay, competitive, non-competitive and uncompetitive. So this is acting out uncompetitive. Uncompetitive inhibition binds to, they basically binds to your enzyme substrate complex. So uncompetitive inhibitor does not, there is no structural similarity between the substrate and it does not have any affinity for the free enzyme. So they binds to the inhibitor of enzyme substrate complex. Allosteric inhibitors change the substrate saturation curve towards the right. So here Km increases but Vmax remains unchanged for aspartate, transcarbonylase and phosphoprotokinase, whereas Km or substrate affinity remains unchanged but Vmax is decreased in case of acetyl coenzyme carboxylase enzyme. So moving to the next question, an on-catalyzed reaction. An on-catalyzed reaction, that means there is no catalyst is used. So here catalyst, we are talking about biocatalyst or enzyme. We are talking about enzyme. Okay, so if there is no enzyme is involved, so an on-catalyzed, high activation energy, low activation energy, balanced activation energy, all of the above. So when enzyme is not involved, the activation in energy is high. That's why enzymes are basically act by lowering the activation energy. Okay, option A is the right answer. So remember what is enzyme. So enzymes are the biocatalyst and they are basically proteinous in nature. What they do, they basically carried out the, or basically increase the velocity of reaction or rate of chemical reaction without being changed inside the, without being changed itself during the overall process. The enzyme word is basically coined by one of the scientists whose name is Kunne, and the enzymes are the specialized protein act as a catalyst. They basically speed up the reaction by lowering the activation energy. So basically lowers the activation energy. So what is activation energy? So activation is the energy it is required to reach at that energy level to form a product. So when the energy reduce this activation energy, that leads to increase or increases the rate of reaction or velocity of the reaction. So moving to the next question, which of the following enzyme has a fibrinolytic activity? So fibrinolytic activity means the ability to degrade fibrin clots, okay. So that means they are basically used as a blood thinner. So they are basically used as a blood thinners, okay. So correct answer is your urokinase. Pepsin is used as a proteolytics. L-asparaginase used as anti-cancer agent. Lysozymes are used as a antimicrobials. So moving to the next question, yeah, you can see antimicrobial. Pepsin is used as a proteolytic. Penicillin is used as antibiotic. Antagonize the action of penicillin. L-asparaginase is used as antileukemic. Hyaluronidase is used as a mucolytics. Okay, so moving to the next question, the fat soluble vitamin is. So you know this is one of the important questions. So how to remember this? So you have to remember the enzyme classification. So vitamin classification. So vitamins are broadly classified into two categories. One is your fat soluble, another one is your water soluble. Fat soluble enzymes are vitamins are A, D, E, K, whereas water soluble are again divided into two part, B complex and non-B complex. B complex again divided into two subclasses. Non-B complex is vitamin C. And non-B complex again divided into energy releasing and hematopoietic and energy releasing. Energy releasing example is your B1, B2, B3, B5, B6 and B7, and energy releasing is your B9 and B12. Okay, you can see the classification over here. Yes, this is your fat soluble vitamin, your vitamin A, D, E, K. Water soluble again divided into non-B complex and B complex. Non-B complex example is vitamin C. B complex example is energy releasing and hematopoietic. Energy releasing example is your thiamine, riboflavin, niacin, pantothenic acid, pyridoxine and biotin, whereas hematopoietic is folic acid and cyanocobalamin. Next, which of the following disease is caused by niacin deficiency? Niacin is basically nothing but your vitamin B3. So niacin deficiency leads to rickets, night blindness, scurvy or pellagra. Yes, which is the correct answer? So niacin deficiency is pellagra. Okay, so rickets is due to vitamin D deficiency in children. Vitamin D deficiency in children. Okay, night blindness is due to vitamin A deficiency. Okay, so next is your pellagra. Pellagra is due to vitamin C deficiency. Pellagra is due to vitamin B3 deficiency. Vitamin uh, vitamin B3 deficiency, and scurvy is due to vitamin C deficiency. Okay, so you can see vitamin B3 and niacin deficiency. Niacin and nicotinic, also known as pellagra preventive factors. So this is the coenzyme is NAD and NADP. Its deficiency leads to pellagra. This disease is associated with dermatitis, diarrhea, dementia. If not treated, that leads to death also. Okay, so moving to the question number 7, the active form of riboflavin. Active form of riboflavin, whether it is FMN or FAD, NADP or NADPH, AMP or ATP or retinine. So remember, NAD or NADP is the active form of niacin. ATP and you are the energy currency. FMN and FAD is the active form of riboflavin. So option A is the correct answer. So option A is the correct answer. Okay, so moving to the next question. This table tells about the active form. Thiamine, the active form is thiamine pyrophosphate. Riboflavin, the active form is FMN and FAD. Niacin, the active form is NADP plus and NADP. Pantothenic acid, the active form is coenzyme A. Pyridoxine, the active form is pyridoxal phosphate. Cobalamin, having active form methyl cobalamin. Folic acid, having active form tetrahydrofolic acid. Biotin, having active form biocetin. Ascorbic acid is itself an active form. Okay, so moving to the next question. Total energy gain from one molecule of glucose under aerobic condition. Total energy is gained during aerobic condition, that is 8 ATP, 38 ATP, 12 ATP or 36 ATP. Total energy gain from one molecule of glucose under air. So your correct answer will be 38 ATP. So total 38 ATP will be produced during aerobic condition of glucose. So glucose converted to glucose so you can remember like this. So you can remember like this. So total ATP produced during glycolysis is 8. Total number of ATP that formed from TCA cycle is 24, and 6 ATP is formed during conversion of pyruvate to acetyl coenzyme. Okay, so what happens during glycolysis? See what happens. Your glucose converted to pyruvate. This is during glycolysis. Here 8 ATP will be produced. Now this pyruvate converted to acetyl coenzyme. Here 6 ATP produced. Now this acetyl coenzyme undergo TCA cycle. Acetyl coenzyme undergo TCA cycle. So how many ATP will be produced? Total 24. So if we will add 24 plus 6 plus 8 is equal to total 38 ATP. Total 38 ATP can be produced. So moving to the next question. Galactosemia is mostly due to the deficiency of which enzyme? Galactosemia. Galactosemia is basically deficiency due to glucose 6-phosphate, HMG coenzyme reductase, galactose phosphatase uridyl transferase, या L-glucolactone oxidase. So this is basically due to your galactose phosphate uridyl transferase. You can see galactosemia is a condition in which body is unable to use sugar called galactose. So galactose is a part of main sugar found in milk, which is called as lactose. It is found in milk is called as lactose. Galactosemia can be problem for newborn because galactose is present in both breast milk as well as baby formula. Okay, that's why this is such type of condition can be seen. So classic galactosemia occurs when the enzyme galactose 1 phosphate uridyl transferase is missing or non-functional. So moving to the next question, glucose 6-phosphate required which metal for activity? Which metal is required for increase the glucose 6-phosphate? So first step is, so this is the first step of glycolysis. So that means your glucose converted into glucose 6-phosphate, glucose converted to glucose 6-phosphate in the presence of enzyme hexokinase. In the presence of enzyme hexokinase, here which ion, here 1 ATP will be utilized, 1 ATP will utilize, and the ion that required is your magnesium ion is required. So option B is the right answer. Okay, so you can see, yeah, this step. So moving to the next question, normal safety range of potassium in adult blood. So what is the normal health range of potassium in a adult blood? So it is 1.5 to 2 milliequivalent per liter, 3.4 to 5 milliequivalent per liter, 6 to 8 milliequivalent per liter, or 0.5 to 1.3 milliequivalent per liter. The correct answer is 3.4 to 5 milliequivalent per liter. You can see potassium is the major ion that is responsible for the different physiological conditions. So the normal level should be 3.4 to 5, and higher level of potassium can lead to hyperkalemia. Okay, so banana is the rich sources for the potassium. Okay, next is Itai-itai disease is associated with which atom? Itai-itai disease is associated with cadmium, mercury, lead, or iron. So Itai-itai disease is basically associated with cadmium. Okay, so Itai-itai disease is caused by cadmium exposure. It produces a result from activity leads to industrialization, and this condition was first recognized in Japan in 1960. Itai-itai disease is characterized by osteomalacia. There is a severe loss of calcium with a severe bone pain associated with renal tubular dysfunction. Kidney problem also seen. Okay, Itai-itai disease is mainly affected women residing in rice farming areas irrigated by the contaminated Jinju River in Taioma, Japan. Okay, this Itai-itai disease is associated with higher exposure of cadmium to humans, so that can lead to your osteomalacia, renal tubular dysfunctions, along with a severe bone related problem. Okay, along with severe bone related issues. So moving to the next question, what primarily regulates the ingestion of water in response to change in plasma osmolality? What primarily regulates the ingestion of water in response to change in the plasma osmolality? Your hypothalamic control center, kidney function, liver function, yeah, pancreatic hormone. So basically your hypothalamic control center regulate this activity. So you can see the explanation. Yeah, so water intake, water is supplied in the body by exogenous or endogenous sources. So exogenous sources such as ingested water, beverage, water content of the solid food constitute the exogenous water sources. Water intake is highly variable in many range from 0.5 to 5 liter. It is largely depend upon the social habit and climates. In general, people living in hot climate drinking more water. However, ingestion of water mainly controlled by our thirst center present in the hypothalamus. Endogenous water is produced by the metabolic water is produced by the endogenous rate. So around 300 to 350 ml of water is derived from this oxidation of foodstuffs. Okay, so moving to the next question, according to the given data, how much water is obtained from a foodstuff? So around 300 to 350 ml of water is basically derived from a foodstuff daily. Okay, 300 to 350 or 700 ml approximately. Okay, so you can see drinking water and beverage 1500 ml, foodstuff provide 700, metabolic gas foodstuff will be provide around 700 ml, metabolic water provide 300 ml, water intake around 25 to 2500 ml. From this your urine will be 1500 ml, skin 450 ml will be excrete, lungs 400 ml excrete, feces 150 ml. Okay, so remember foodstuff 700 ml, metabolic water 300 ml. Okay, I hope all of you are understanding what we are discussing. So moving to the next question, which of the following is an example of product that developed through a medical biotechnology? Antibiotics, biofuels, genetically modified crops, या industrial enzyme, which is developed through the medical biotechnology is your industrial antibiotic. Always or industrial enzymes can also be the answer. So medical biotechnology has led a development of various pharmaceutical products including antibiotics, vaccines. Okay, these are includes your antibiotics, vaccines, therapeutic protein which is used for the diagnosis, treatment and prevention of the disease. For suppose your natural product or synthetic compound can be developed and that can be upon large scale produced, and that can be done ADMET study or PKPD study for animals or cell for the further checking its activity against different species of microorganism. So moving to the next question, which of the following technique is used to cut DNA at a specific sequence? Which following techniques? So polymerase chain reaction, gel electrophoresis, restriction enzyme, yes, Southern blotting. Yes, which technique is used? So if we'll see, this gel electrophoresis is done for the transfer of RNA proteins. Southern blotting is for DNA. Polymerase chain reaction for amplification, but these restriction enzymes are basically used for the cut the DNA at a specific site. Okay, this restriction enzyme cut DNA at a specific sequence, and they basically that cutting can helps us to manipulate different gene or different genetic gene sequence into that DNA fragment, which is further used for the genetic engineering technique. Okay, this restriction enzymes helps us to develop more type of product with the help of genetic engineering. Okay, so moving to the next question. Yeah, the functional unit of kidney. So functional unit of kidney is what? So functional unit of kidney is nephron. So you can see if it is a structure of kidney. So our kidney is present. Okay, they are collected with the two bladder. Okay, kidney has two part, one is your cortex, another one is medulla. So between this there is a nephron is present. Yes, nephron is present. That nephron is considered as the functional unit of the kidney. Okay, so what is the function? So nephron helps in what? They helps in filtration. Okay, they helps in filtration. So you can see nephron is a functional unit of kidney responsible for the filtering blood and forming urine. Each nephron includes glomerulus which filter blood and tubules where the reabsorption and secretion takes place to produce urine. While the glomerulus is a part of nephron, it alone is not a functional unit. The corpus and calyx are the other part of the kidney that involve in the filtering process, collection process, but does not function independently as a nephron do. So moving to the next question, what does SGOT stand for? Serum glutamate oxaloacetate transferase, serum glutamate oxaloacetic transport, serum glycine oxaloacetate transaminase, या serum glucose oxaloacetate transport. So SGOT stands for serum glutamate oxaloacetate transferase. Okay, SGOT stands for serum glutamate oxaloacetate transferase. So this is an enzyme. This is also known as aspartate transaminase. This enzyme convert aspartate into alpha ketoglutarate and into oxaloacetate and glutamate. SGPT, these are serum glutamate pyruvate transaminase. It is also known as alanine transaminase. This helps in the convert alanine to alpha ketoglutarate and pyruvate and glutamate. Okay, these two enzymes are the important enzyme, are the important biomarker for the liver disease. So during different type of liver disease, so their level significantly increases, and these are basically act as a diagnostic tool for the identification of different type of liver disease. Okay, so moving to the next question. Which type of anemia is a genetic disorder and that result in abnormally shaped rare RBC, iron deficiency anemia, megaloblastic anemia, aplastic anemia, या sickle cell anemia. Remember, sickle cell anemia is a abnormally shaped red blood cells is produced. So let's learn. So sickle cell anemia is anemia that inherited from the inherited form of anemia in which RBC became rigid and shaped like a shrinked or shrinks. These abnormally shaped cells can get stuck in a small blood vessels and causing pain and impairing in blood flow. These you can see they get accumulated and that prevent the blood flow, and that leads to pain in the bone, pain in different region. Okay. And next question is, which of the following blood test that is used to assess the kidney function by measuring the waste product in the blood? Hemoglobin, blood urea nitrogen test, platelet count, या white blood cells to measure the kidney functions. So blood urea nitrogen is basically used. You can see blood urea nitrogen, that basically what they do, they basically measures the amount of nitrogen in the blood that comes from the waste product as a urea. It helps to evaluate the kidney function, since the kidney is responsible for the removing urea from the blood. So this blood urea nitrogen test, it is one of the important tests for the assessing the kidney functions by measuring the urea level in your blood. Okay, I hope all the questions that we have discussed today is clear to everyone. If you have any questions, so you can put your question in the chat box, so we will try to answer your queries. Thank you so much for attending my class. We'll meet in the next class. Thank you.